Hello, everybody. I'm going to just tell you what our plan is for the agenda today. We're going to try and keep this at 30 minutes, and it's a momentous day. We're really excited that everybody is here to enjoy with us this big, exciting day for everybody at UW Medicine and UW. So we're going to kick it off with a welcome from Anna Kasi, UW President, followed by Tim Dellett, CEO of UW Medicine, and then we're going to hear from David. We would like to have questions at the end of the press availability without interruptions if we can. And then can we make sure and turn off all our phones so we don't have any vibrations or anything. And then we will be going over to David's lab to get some video and photos of him greeting his faculty and staff there. And you can follow us. It's about a block, so it's walkable. And if you need to leave some of your equipment here, you can do that. We will have people here that can watch that for you. Thank you. And we're going to thank you, Anna for welcoming everybody.
Let's be honest, this is about as good as it gets. I mean, just no question about it. This is as good as it gets. And, you know, good morning. This is truly an extraordinary event. Although in the life of David, I think that, you know, prizes like this are kind of getting ordinary. And that is such an incredible tribute to him. You know, we are so proud to be the home of some of the most remarkable, creative, innovative, and talented faculty, and we don't have a better example of that than David. And they are the reason why we are recognized as one of the top public universities of the world in terms of impact. Because this is, if you look at David's work, this is about taking these great basic science ideas and pushing them out so that they make a difference in the world. And that's what we are all about and what we want to be for the state and for the world. I was teasing earlier that he is a fabulous local example. And what I mean by that is he's a local kid, Garfield High. I mean, he really is a local kid, and he's having a global impact. No question about it today. And we are so proud to recognize our 8th Nobel Prize winner. Since joining the University of Washington faculty in 1993, David Baker has helped define what modern biology is through a truly visionary approach to protein science. Just by looking at his current roles, you can see the scope of his work. I won't make pretend to understand it all, but I do understand the impact that it's having. He's a professor of biochemistry in our School of Medicine, and he's an adjunct professor in genome sciences and bioengineering and chemical engineering and computer science and physics. And that is because the scope of his work is so broad. And that's what we're trying to do here, is make the barriers to this kind of interdisciplinary approach as low as possible. He's also the director, of course, of the Institute of Protein Design at UW Medicine and a Hughes Medical Institute investigator. He's also an elected member of the National Academy of Sciences. He's had a bunch of awards. We could be here all day if we talked about all of them, so I'll just mention a few. The 2021 Breakthrough Prize in Life Sciences. Just earlier this year, Times named him one of the world's 100 most influential people in health, a list he would have made if it was the top 10 most influential people in health. He won the Frontier Prize of Knowledge Award in biology and biochemistry and biomedicine by the BBVA Foundation in Spain. And earlier this month, he was named a Clarivate Citation Laureate in recognition of the importance of his research papers through scientists around the world. I want to just stress that what we're talking about is that other scientists are citing his work as being critical to their work. And that is what it's all about. We couldn't be prouder of this lifelong Seattleite. His parents were faculty members here at the UW, and this graduate of Garfield High, which is just about as good as it gets. Dr. Baker, on behalf of the University of Washington, congratulations. And But not just congratulations, thank you for making our university and our world a better place. And you're just beginning. I mean, that's what I want to stress. Nobel Prizes often come at the end of someone's career. David has his career ahead of, this is just the beginning. And so now I'd like to introduce Dr. Tim Dellett, CEO of UW Medicine and one of the strongest supporters of David across our campus. He's also the Paul G. Ramsey Endowed Dean of the School of Medicine. Tim, you understand his work better than I do, so take it away.
Well, thank you, President Kause, and I'm just so happy for David, for his family. It's great to see Hanla here, who also is a star in her own right, for our entire University of Washington community, and especially the community of protein design, the Institute of Protein Design, and all the individuals who have been involved and worked with David throughout his career. You know, in my roles, I really have the privilege of helping to support and often facilitate the way for our faculty, our staff, our trainees, our students. And our faculty really conduct world-leading biomedical research that expands our scientific understanding and leads to new frontiers in science and medicine. And this is just a phenomenal day. It's a wonderful recognition of really the lifelong work of David, and it's great to have this come while David's still in his prime, and can't wait to see what he continues to discover. You know, David and his team really contributed and led the cracking of the code to protein structure. How amino acid chains fold together in a three-dimensional structure to be the building blocks of life. By harnessing artificial intelligence and computational protein design, you know, the Institute for Protein Design, now they create novel proteins that have never been seen before in nature to really help us address some of the most challenging issues we face, whether it be in medicine, technology, or even sustainability. Among some of the most promising applications for medicine, protein design is paving the way for development of new and highly effective vaccines, novel therapies such as antivirals to block viral infection, treatment of celiac disease, cancer immunotherapeutics. It is just phenomenal what David's scientific innovation and discovery is leading to in terms of impact for health of our communities. Protein design also has the potential to provide, again, innovative solutions to some of our biggest environmental challenges by creating new biodegradable materials, sequestering carbon, and breaking down pollutants. As director of the Institute for Protein Design, and as President Kause mentioned, David's impact goes far beyond his research here at the University of Washington. It truly extends to the global scientific community through over 600 published papers and his real commitment to open science that is available freely to everyone. It includes moving research to practical applications more quickly through 21 companies that he has co-founded and more than 100 patents. So again, taking that scientific innovation, that discovery, and translating it to real-world impact. It also includes his role as a mentor to more than 90 students and postdocs who are now making their own contributions to the field of protein design in faculty positions around the world. So thank you, David. Thank you for your amazing contributions, not only to our community here at the University of Washington, but really globally. And so now it's my distinct honor to introduce our newest Nobel laureate at the University of Washington. Please join me in welcoming Dr. David Baker.
Well, what can I say? I want to start by thanking almost everybody here, and so I want to thank Anna Marie and Tim for really making the University of Washington an absolutely wonderful place to do science. As Anna Marie said, it's an incredibly collaborative place, and I've never felt during the whole time I was here that there were really any boundaries within the university. And so I'd like to thank both of them for making the university, the university leadership has just been amazing.
Kind of a big boundless t-shirt.
And, you know, I've been here for quite a long time, and I never thought for a nanosecond about leaving. So it's really been great privilege to be here. And so keep up the great work. And Tricia is here, and she really played a huge role in everything we've been able to do in the founding of the Institute for Protein Design. And my wife Hanala is here, and she basically made everything possible by, among other things, putting up with me over the years. And Linda, our director of the Institute for Protein Design, is really kind of the adult in the room and making sure that everything happens, and while I just sort of play around. Let's see in the back, I think I see there my colleagues Frank and Neil, who've it's been great to start the Institute for Protein Design and have some of the most amazing scientists I've ever worked with. Continue as professors doing really absolutely amazing things, and it's been an absolute privilege to work with them over the years. And as Tim and Anna Marie says, I'm very, very excited about the future. I think protein design has huge potential to make the world a better place, and I really do think we're just at the very, very beginning.
Can you briefly explain what protein design is, why you got into it, and can you talk about your work that, specifically your work that won the Nobel Prize?
Yes. So proteins are the miniature machines that carry out all the important jobs in our bodies and in all living things. So basically all the remarkable properties you see of any animal, plant, or anything, those are being mediated by proteins. Up until, I guess, before our work, the only proteins that we knew about were the proteins that we discovered in nature. So a lot of molecular biology and a lot of biology over the last probably 100 years has been about trying to find, if you're trying to study some biological process like how muscles work or really anything, it's been about identifying what the proteins are. And so sometimes you might read in the newspaper or elsewhere about some new exotic protein that someone has discovered that has an exotic name. And those were really all the proteins that we knew about, were the ones that sort of came through evolution. And I kind of liken it to sort of the Elven runes that you discover from prehistory, because they're kind of all have these exotic names. And the idea that you could make new ones was kind of a crazy idea. So because proteins solve all the problems in that living things have to deal with so well, if you could make new proteins, you could potentially solve a lot of current problems for which there aren't proteins to deal with. Like, for example, there are new problems that arise because we live longer today. So, diseases like Alzheimer's are important. Cancer is more important. We're heating up and polluting the planet, so we have new ecological problems. So if we had a long time to wait, then maybe new proteins would evolve to help deal with these problems. But evolution happens on, geological timescales. So with protein design, now what we've learned how to do is to design completely new proteins that have new functions. And so now we're working on problems like designing proteins to attack cancers and acting much more specifically and precisely in the body, so be safer and more effective than current treatments. Similarly, for autoimmunity, being able to dampen down the immune system where it's gone awry. My colleague Neil, who's in the back, has developed the first de novo designed medicine that is in use in humans, a coronavirus vaccine. And his group is making great progress to a universal flu vaccine, for example. Outside of medicine, I should say another area that is important in medicine that we're working on, in addition to vaccines, are proteins that will neutralize pandemic viruses. And so we're sort of going through the list of the greatest viruses of concern and designing proteins to block them. and some of these are headed for human clinical trials. Outside of biology, I mentioned that we're polluting the planet. We're putting a lot of plastic out there and a lot of other toxic things. We're designing proteins that carry out chemical reactions to, for example, to break down plastic molecules and other pollutants that we're putting into the environment. We're designing proteins that interact with solar radiation to try to increase the efficiency of photosynthesis. And we're designing proteins that to mediate enhanced carbon fixation. And we're just starting to collaborate with others at the UW to work on the methane sequestration problem. And there are also huge opportunities for proteins in making new types of materials. We're familiar with things like bone and tooth and shell, which are made from proteins interacting with minerals. And now with protein design, we can make proteins that mediate mineralization and create new interesting materials from not just things like calcium carbonate and calcium phosphate, but things like semiconductors. So there's a lot of different applications now. I'm not sure if I answered everything you asked, but you got a mouthful anyway.
Yeah, that's what I was hoping for.
Yeah.
Can you talk a little bit about what it was that first drew you to this work? At what age? I mean, that your parents were faculty members here, but can you talk a little bit about their influence on you in becoming a scientist, as well as what specifically drew you to?
Yeah. So the problem, I think what really interested me about the problem in the first place is that so living things are very different from inanimate things like the rest of chemistry. You have chemicals which are not alive, and then you have biology things which are alive. And proteins are kind of at the intersection between the two, because a protein is just a very large molecule, and so it's very much sort of a chemical sort of thing. It's just a molecule with made out of atoms connected by bonds. But proteins have this amazing ability to self-organize. They fold up into shapes that kind of have these magical functions. And so it's really kind of the simplest case of biological self-organization. And I think it took my path to getting to being interested in proteins. Actually, it was kind of a long and circuitous path. I didn't really start working on proteins until I became a professor here, really, which so it wasn't something that I even was aware of when I was growing up. So I have to thank my parents, who are actually going to be at the celebration later, for teaching me how to think and do many things. But the protein part came really kind of late, and it wasn't like I was fascinated as a kid by proteins.
But what was it? At what point did you start realizing, wow, this would be a fascinating topic?
I think, well, I actually in college, I went to Garfield High School, and then when I went to college, I actually thought I wanted to be a social studies and then a philosophy major. So it was only kind of late that I switched to biology. In my last year of undergraduate, my fourth year as a senior year, I took a biology class and learned about proteins and how they fold up. And I thought that would be a really interesting thing. It was really interesting, but I remember you had to write a paper in the class on something. I wanted to write a paper on that, and the TA said, "Oh, that's way too complicated. No one will ever understand it." So I didn't do that. And then, but I got excited about biology, and then
another university with other TAs.
And then I got excited about biology, though, and I went to graduate school and worked on something completely different. And then, like I said, it was really when I came here that I started working on the protein folding problem, and then that got us into how to design proteins. And again, I have to thank the University of Washington, the Department of Biochemistry, for kind of taking me in the first place, even though I didn't really know what I was doing. And then I had the freedom really here to pursue the science as it developed. And I really, it wasn't like I had a plan. I've always said I can never see more than 3 months ahead. So, yeah.
I understand from talking to your colleagues that you have some skeptics early on that this could be done.
Yeah.
Can you tell us why they thought that couldn't be done and how you surmounted?
Yeah. Well, to begin with, the only proteins, like I said, that were known were the ones that came through the evolutionary process. So saying you could make a new protein, someone could sort of interpret like, oh, I can make a new animal, or I can make a new plant. And those would be obviously really, really hard to do. So you're kind of make something that's kind of like a living thing, but you're making it from scratch. I mean, is that really possible? And then after we showed we could make proteins that folded up, but were totally inert, the idea that you could give them functions and make them do things was kind of a crazy idea. And we were actually kind of on the lunatic fringe for many, many years in saying that we were going to, in trying to make proteins that could carry out very sophisticated functions, just starting completely from scratch. So many people, many engineers in biology have taken proteins that already exist in nature and tried to modify them a little bit to solve a problem. And what we've been trying to do is just start completely from scratch. And so people said that was a crazy thing to do, and no one thought it was very. It was very much on the lunatic fringe. Until when we start getting better at it, and really this is in the last several years, suddenly it's in the mainstream. And, you know, companies start all the time and say, we're part of the protein design revolution. And, you know, so it's kind of interesting with this field going from being something which was really kind of crazy and didn't seem like it would ever be useful to anything, for anything, to being something that's so mainstream that big companies, you know, are just starting all the time, and it's kind of on the tip of everyone's tongue.
Can you share what may be next in your work, or what you'd like to accomplish next?
Yes. Well, you know, as was already alluded to, I think we're really just at the beginning. We figured out how to design new proteins. We figured out how to make them, you know, bind to cancer targets, for example. And we're getting better at learning how to make them catalyze chemical reactions. So we've really learned a lot about how to design proteins with new functions. So I think now what's tremendously exciting is to look at the vast array of problems that humans face and try and design proteins that solve these problems. And I'm really fortunate. I hope that many of you can come over to the lab in just a moment. You'll see there are large numbers of super brilliant, super smart, super energetic students and postdocs who are now coming from all over the world to the University of Washington to learn how to design proteins, and each one of them has a problem they want to solve. For example, Susanna, who came from Mexico, is designing proteins that block the major components of snake venom as a better anti-venom. And you'll also meet students who are working on improving the efficiency of photosynthesis. So really across the board, there's so many exciting things to do now. So I really do think we're just at the beginning of the impact that we can have.
And is there a single problem that you see kind of rises to the top? You mentioned a few of some of what the students are doing, but is there one that, gosh, if we could just solve this problem, what would that be?
You know, I kind of love them all. I mean, they're all, I mean, we only work on things that I'm really, really excited about. And I think the neat thing about this area now is there's just so much potential across the board.
Do you have a dream function for one of the proteins that you could create that would be considered lunatic fringe today? Yeah,
that's a good question. Well, one of the things that that exists in biology are molecular motors. So, for example, the way that our muscles move, they're little machines that are involved in the contraction and the movement, and the way that things get organized in our bodies and in our cells is through these very sophisticated molecular machines. So, and what they do is they take a chemical fuel and they break down that fuel just like kind of a car. They take a fuel and they use it to do something very actively. So the newest generation of projects in my lab is to design molecular machines. Now, what could they be good for? Well, in biology, they're machines inside our cells that do quality control and are important for removing a lot of the junk that happens during cellular transactions. There really aren't any machines outside of our cells in circulation. But we know that there's all sorts of, you know, junk accumulates in our bloodstream. We get aggregates, we get amyloids forming. So if we could make machines that could go around in the blood and in circulation and basically do cleanup and help remove toxic tissue, that could be a whole new way of treating disease. So that's one of the sort of more lunatic-y fringe that we're working on, things we're working on now. Another, I'll give you one more example. One, and that is, I think I already alluded to this, but we have tooth and bone in our bodies, and there's shells, and those are proteins interact with minerals. What if we could make completely new types of materials by designing proteins that interact with other minerals and cause, and you can imagine all kinds of hybrid materials that could be superior in many ways to any of the materials that we have today, including semiconductor materials. So it could be a new way of patterning things for electronics. I'd say that still qualifies as lunatic fringe also.
Do we have any questions before we head over to his lab? Any last question?
I just want to make one last comment, and that's that David's achievement isn't winning the Nobel Prize. The Nobel Prize is a recognition of David's achievements thus far. The best is yet to come.
Thank you. Thank you.